If I can take a minute and just say a few words.
Sure.
First of all, thank you very much for joining us today. We have a very distinguished speaker, and I'll say a few words, but I also want to welcome our colleagues at NSF. Mitra is online, looks like, and Sylvia is online too. And we're very happy that you can join us. Without taking more time, I want to just say, how delighted we are to have Rita speak. We had Simon and his students speak last time, and this time the second talk is by Rita. So we couldn't have had a better start to this Expeditions Colloquium series. I don't think so anybody here has not heard of Rita's work, but just in one sentence, she's absolutely an amazing scientist and a great motivator for scientists all around the world with exceptional work. I mean, she's just remarkable career spanning over 50 years, I think, Rita is, if I'm correct. It's a testimony to the work and your ability. So I'll let you take it from here. Thank you very much for doing this. Thanks again to you and Simon for starting this off. We couldn't have a better start to this series.
Well, thanks you very much, Madhav. It's terrific to work with you and with Simon. I think the whole team that you've pulled together is just amazing, and it's exciting and fun. What I'm going to do is initially I just want to cover some background information because I think it's important to lay the groundwork in which I'm doing some current COVID work that I think is very relevant to the whole team that we've pulled together. I want to talk about the environment and its role in controlling and influencing infectious diseases. That's essentially one of the major themes of our whole program. What I'd like to focus on is on waterborne diseases because waterborne diseases, I think, are fundamental in understanding infectious disease. We're currently working on air transmission as well, but I'll get to that at the end of my talk. The work I've done is mostly on the diarrheal diseases because they're incredibly common, and cholera is, in fact, the continuing pandemic. We have a new pandemic, which is lethal, the coronavirus, but we've been dealing with the cholera pandemics over waves of outbreaks, and the numbers are in the billions, and the deaths are in the 200,000 a year globally. So when you look at the other kinds of infectious agents, when you have contaminated water, there are about 25 or 26 diseases transmitted by contaminated water, for which we have really only a couple of vaccines. One is the cholera vaccine, which is only about 60 to 70% effective, and usually has to be repeated every couple of years, plus it requires a cold chain. The other is rotavirus. It's very expensive, and that does require a cold chain as well. So we haven't gotten very far in dealing with the problems of safe water. So cholera is a global disease. It's an acute water-related diarrheal disease. It's globally transmitted, and it occurs in about 50 countries. Every year, at least 7 million people. We've unfairly referred to the Bengal Delta as the native homeland, but that's not really fair because we had cholera in Baltimore, Maryland, in New York, in the US up until about 1920 when we began chlorinating and distributing chlorinated water to households. But the discovery that my team made, good Lord, it's now 40 years ago, was that the bacterium is naturally occurring. It's an emerging infectious disease. It's naturally occurring in the environment. It carries out a variety of very important ecological functions, cycling of carbon and nitrogen. So we can't ever eradicate it, but we surely can control it. The work that I will be described was begun in the Chesapeake Bay back in the late 60s, and the numbers indicate the stations that are still being monitored even today. They were established by Johns Hopkins, and we used their catamaran in the early days flying the Chesapeake. But we were able to isolate Vibrio cholerae from the environment. And that was important because it made a major discovery that it has a host. The host is a copepod. It's a plankton species, and the bacterium is a rod-shaped curved rod with a polar flagellum, and it is associated with the gut and the surfaces on the carapace of the copepod. All Vibrios carry a chitin digesting enzyme. We developed very quickly a very crude model. This was, 40 years ago. Simply showing that in the warm spring months, when the sunlight increases, the algae and the phytoplankton bloom, followed by the zooplankton, which blooms as it feeds on the massive phytoplankton populations. And then as the food supply deteriorates, the zooplankton bloom crashes, and that releases lots of vibrios into the water column, and then transmission, usually in the late summer or post-monsoon, of cholera occurs, let's say in Bangladesh. And then with fecal shedding, we regenerate the cycle. So that's the crude initial model. Now we were challenged by, say, by those who didn't really endorse our hypothesis that was maybe just Chesapeake Bay. So we began studies in 1975 in Bangladesh, working in the Sundarbans. This is the southern tip that feeds into the Bay of Bengal. And there we were able to do work that allowed us to show that indeed there the copepod serves as the vector, the host. It's a vector-borne disease. It's just that the copepod swims and it doesn't fly, but otherwise it's similar to malaria, which has a mosquito as its vector. Now, of course, person-to-person transmission occurs. We can't deny that, but it's the environment that's the source of the bacterium. And so that completed the cycle and allowed us to say that this is really a global phenomenon. So the next thing we did was to consider the possibility, since at that time in the early 1980s, early 1990s, Landsat had been launched and was measuring chlorophyll, sea surface temperature, sea surface height, a variety of other parameters. And it occurred to us if there's a relationship with phytoplankton, which carries chlorophyll, and if we could calculate and adjust for the zooplankton bloom, we could actually use satellite sensors to predict and monitor the epidemics, particularly in Bangladesh. So in this case, in 1985-86, that's 25 years ago, we collected the data in the Sundarbans of Bangladesh, and then we worked with NASA, and we looked at a quadrant just off the Sundarbans in the Bay of Bengal for the sea surface temperature, sea surface height, and chlorophyll. And then we did the calculations. We published this in PNAS 20 years ago, showing that indeed satellite sensors work very, very well for being able to detect and predict at least this infectious disease, which has this host that can be monitored by satellite sensors in the system that NASA had launched. The whole idea now is that we have a powerful tool, and we've much improved it to the point where we can determine if there's a heavy rainfall, there'll be about a 7% increase in cholera cases. If there's a 3 or 4 millimeter increase in chlorophyll per unit, there'll be about a 33% increase in cholera cases. So the point is that we should sharpen prediction and intervene early to prevent cases and provide treatment, rather than doing what too often is done by the public health machine, is to go in when it's at its peak, and by that time you're really sort of there when the horse has left the barn, so to speak. So working on prediction, we provided a more detailed understanding of cholera. And we now know cholera operates really in 3 modes. There's the epidemic mode that occurs internally, let's say in Dhaka or in India, in Delhi, which is internal. There's a river system there, as opposed to the endemic, which is along the coastal area of Bangladesh. There's cholera really pretty much throughout the year, but it has major peaks in the spring, which correlates with the plankton bloom, and again with a subsequent bloom in the fall. And so those peaks are intervened by the monsoon, but it's a very predictable year after year. So, and then there's a mixed mode where you have occasional epidemics, but kind of a low-grade endemic cholera. Now, a colleague, and I think Antar may be on the call, he's joined our team, working with us in the team that you're running, Madhav. We've improved the prediction, and we've expanded the use of satellite sensing to the newer satellites that measure precipitation, temperature, movement of populations, because that allows, again, a much more sensitive prediction of cholera. We've reached a point where we can actually, 4 weeks ahead of time, we can predict the high risk. This is the model that we now work with and the various satellite systems. Going back to the early data in India, which a colleague, Dr. Elizabeth Whitcomb, a remarkable individual, who was running a clinic in London, and interested and had done a PhD as well on the waterborne diseases of India along with her MD. She had discovered these maps in India, and we had gone to the maps. We were able to transfer the data from about 1873 to about 1923 or 1930, where the British colonials had recorded every death from cholera and malaria. And we were able to get this into the computer. So now our sensitivity, coupled with very, very precise measurements of the various satellite sensors, allows us then with this model, which we now have determined from the India data, that when you have 2 or 3 months of very hot weather, followed by a very heavy rainfall, and in an area with water insecurity, that is very poor infrastructure, or as in the case of Haiti, damage become from the earthquake, then there's a very high risk of cholera. And this allows an early warning system to implement with physicians and medical supplies, etc. So what we had done was in 2017, we did a retrospective analysis with our much improved and more sophisticated model. And indeed, our prediction of risk, which is the blue, green, red area, when we then map the actual cholera cases for 2017 in Yemen, it was bang on. It was really a remarkably good coincidence of the data. We published a quick paper. It was picked up by Scientific American, and a little squib, a paragraph on this new way of using satellites for predicting cholera. This was read by a British scientist who works for the Department of Foreign Aid in England. He called us in January of 2018 and said, hey, let's work together. You tell us where in Yemen in 2018 the highest risk of cholera will be, and then we'll locate our supplies and our personnel in those specific areas. And that we did, and we were able to reduce cholera significantly. In fact, it got an award for our colleague in England, and we ended up on a BBC program. And one of the interesting things, we've continued. Now we provide for UNICEF and for the British Aid Agency with the assistance of the British Meteorological Society Department and NASA. We provide 4-week early prediction of risk, and we're getting close to being able to predict 8 weeks ahead of time. But this was kind of an added benefit in the team that Antar has put together. Did a survey of Sudan, and we picked up risk of cholera. They didn't expect it, and we notified the British aid agencies, and indeed cholera did appear in Sudan in 2019. The UK aid experts are using the predictions, and what they do is stockpile hygiene kits, jerry cans, and chlorine to treat the water. And this has proven very, very effective, and it's ongoing, and we've been asked to extend this to Africa. We'd already done some work in Mozambique and Senegal, and now we're expanding into some other countries of Africa where cholera is seriously epidemic. So now, what we thought, beginning in January, February, was that we had this extremely powerful model and this effective prediction capacity. Could we modify it for COVID-19? And so going back and looking at data on particularly aerosol transmission and humidity and moisture, droplet transmission really, we found that indeed there was a peak for the SARS epidemic, and COVID is really a SARS variant, it's a coronavirus. And so that has led us to try this out. And sure enough, looking at the US, a preliminary study or analysis we did of the aerosolization index. The deep blue shows the high human cases, and on the right is the predicted. So this model works in this case because it tells us that the coronavirus is influenced by the environment, and others have been studying the seasonality. But what we think is that we should be able to do a real-time COVID-19 prediction, particularly with respect to the fall season and with our model. This is a the map that we drew in May, I don't have the latest June figure to show, but this is what we did for last month. The other thing that we're doing, which is very interesting, is looking at what I call the ground truth measurement. We're just starting a contract for the state of Maryland looking at wastewater, sewage, for the coronavirus, because with our very elegant technique for extracting DNA and RNA and being able to identify all pathogens, including the coronavirus and its variants, the COVID-19 and the COVID-19 variants, based on the studies that have been done by a number of people, showing that there is a distinct relationship of the victims discharging the coronavirus in the stool. And this study, which was done by a Chinese team of the COVID-19 virus, the red line shows individuals, these will read horizontally, individuals with a positive throat swab. And yellow is the discharge of the virus in the feces. So long after the throat swab is negative, there is discharge of the virus into the feces. The round dots indicate those admitted to the hospital, and the red X's are those individuals who were monitored who died from the virus. So this very clearly indicates that discharge into the sewage system is a strong indicator. And based on these other studies that have been done in Australia, in Arizona, and now we are launching this and we've completed some studies for Prince George's County, for Frederick, Maryland, and also for Fairfax, and we're doing some work with DC Water as well. As a mechanism, first, for the outer gals to be able to predict cases even before they are recognized by physicians or the hospital, because they show up in the wastewater, and also as a measure trend analysis to indicate whether masking and distancing and any other methods are successful. And it also provides the governor of Maryland evidence for staging, opening up the community on science rather than simply wishing for the best. So we're in the midst of doing this, and I think it'll provide us then with a mechanism for ground truthing what we have been able to predict, and so we're putting that together. Now, I think it's important to emphasize that this pandemic of COVID-19, very likely, as others have predicted, and I certainly agree, there'll be COVID-20, COVID-21, whatever. And these are the plagues and the pestilences that have gone all the way back to earlier than the 1300s, and they occur with quite remarkable regularity. So we really, really do need to be prepared for the next pandemic. I've made this purposely brief to have some discussion, but I do want to thank my collaborators, particularly those in Bangladesh and India, and the many students and postdocs and techs and visiting scientists who've been remarkable in working over the last 30, 40, or even 50 years. And I'd like to particularly acknowledge Anwar Haq. He was a student, did his PhD, and went back to Bangladesh to run the labs at the International Center for Diarrheal Diseases Research, and then came back and joined my lab as the major tech, and he's now a professor at the University of Maryland. Hassan is an incredible biotechnician, bioinformaticist, I should say, as well as molecular biologist. And Antar is an engineer, but is extraordinarily capable, and with whom I continue to work with all of these. And Sion Young Choi put together our database and has been incredibly helpful and remarkably brilliant. I just want to close by pointing out, in the words of John Muir, that when one tugs at a single thing in nature, he, and I inserted and she, find it hitched to the rest of the universe. And I think this is a powerful message, because the emerging infectious diseases, these pandemics, are really a result of invading Mother Nature, of denuding the tropical forests, searching for wildlife for food in a burgeoning now 7.5 billion to be a 10 billion populated universe. We really, really need to understand the intricacies of nature and the intertwining of climate and environmental diversity and human health. So with that, I'll take questions, and I hope this fits very nicely, at least I think it does, to the great work that you're getting launched with our team.
Thank you very much, Rita. I'm sure this is the best we can do in clapping, and some people will, I guess, do it virtually using things. But wonderful talk. I think this is exactly what we wanted. Just for some of you who might not know, Rita started the Biocomplexity Initiative when she was the head of NSF, and Simon has taken it a step further. In fact, there's his center, of course, and the one in ASU are all called Biocomplexity Institutes. So for us, our new initiative and center here at UVA is also called Biocomplexity Institute. And so it's a very nice match to your interest, and your last quote really sums up the reason we have started working on this. Chris Barrett, who is online, deserves a lot of credit for his vision to set this up here. So with that, I'll open the floor for questions. I have a few, but I'll let others ask questions.
I just wanted to compliment Rita on a terrific talk. As always, she's an inspiration to us. So I didn't really have a question.
Good to hear from you, Simon. Thank you.
Okay, I think I saw Jeff Townsend next.
Hi, Rita. That was a great talk. Thanks so much for presenting the research you've done. Some data have been presented in various forums that argue that pollution levels are a very important predictor of COVID-19 transmission. So with regard to predicting it, I'm just wondering, do you think there's the potential to disentangle urban location, which has, a lot of, density of people has been a big predictor as well, humidity, pollution. There's so many different factors that have a certain level of correlation. Are we going to be able to deconvolve those for the purpose of prediction?
I really think so. We're working on it. Antar, are you on the call or not? If you're on, you might.
Yeah, he is.
Antar, do you want to comment? Because we're now working on aerosols, and his colleague has developed a really, really elegant virus trapping device. And I do think that we're going to find a very close correlation and be able to separate the parameters. Antar, do you want to make any comment?
Yes. Hi, everyone. So the model is compartmentalized right now. So we have the environmental component, the social demographic component, which has the population density, impact of movement, IDP parameters. And so even in the first prediction map for May, these are parameters were in the model itself. But in our July prediction maps, we are going to compartmentalize and then produce that map. The answer is yes. It's happening.
I think you open up an interesting line of inquiry with Antar on the role of environment. And Jeff asked this question. There's a nice paper by Brian Grenfell in Science on trying to connect this as well. I think the satellite imagery would provide us a huge amount of data. As we discussed, we have some understanding of human mobility data as well through flows at a 5 square kilometer level. And I think combined with environmental monitoring through the satellites with human mobility, and we also have now a good collection through a group, the numbers, at least in the US and most parts of the world, like Johns Hopkins has done. We've done a similar project. So I think we should be able to combine these three things together in a rather interesting research direction if folks are interested.
I totally agree. I think it's pretty exciting using cell phones and mobility measured by just that set of data, along with the satellite data that looks at migrations of populations down to a very, very small, relatively small area. This, I think, allows precision that we just really can take good advantage of.
Right. Maybe I'll ask another question that is related to the environmental sampling work you've started. I think that's interesting as well. And one thought that I have, and actually Simon can say a little bit more, he talked to one of his colleagues who is also doing something very similar. But I think it might be very interesting to do the sampling studies on college campuses because they're dorms and you can essentially do a very targeted analysis of the samplings to get early warning indicators across all these dumps. I think a project that we should really consider starting very quickly is to set this up so that at least at UVA and Johns Hopkins, UMD, Princeton, we could actually start doing this. And I think along with other indicators, of course, swabbing and such, this might produce a very good map of how the disease is going through. Would you agree?
Yes, I do agree. And I think I just saw that pop up from Antar. Are you already starting that in Florida?
That's the reopening plan. So we are installing the samplers in schools, like elementary schools, middle schools, high schools, some churches, and institutes. But I think we are, due to travel restrictions, we are going to be focusing on the Florida area. But I think if we can do a grid type of study where we can know that how people are moving, that would really empower our prediction maps. So I asked a question that if Virginia Tech folks can provide us with like geolocated data sets at a high precision level from Twitter, Facebook, cell phones, then it will become very powerful.
But you're doing aerosol sampling, Antar.
Yes, yes.
I think the question that might have asked was with respect to wastewater and sewage, right?
Yes. Right. And I was thinking the reason I brought it up, and I think Antar, your point is well taken, and we will certainly do that too. So great answer. But what I was thinking is we have at least 14 or 15 different universities represented on our project. So Jeff is, for instance, there at Yale, Simon is at Princeton, we are here, Virginia Tech is around, folks at Stanford. If all of us even cover our campuses, we could have set up a global, at least US university level surveillance system effectively through this water thing, and hopefully through swap too. But I think the results from wastewater certainly merits a lot of thought, in my opinion, Rita, in this direction. Simon can say a little bit more. He has already been thinking as well. Simon, do you have to say anything more to this?
Yeah. There is an effort going on led by a number of my colleagues at Princeton. It's not just the college campus, because we don't have any students around at the moment, except a few graduate students who didn't go home. But it is in the community. Jane Lipchenko and Ben Dalziel are conducting a similar effort in Corvallis. She's not part of our project. I know efforts at Rutgers and elsewhere. I was thinking, Madhav, whether a good thing for us to do might be to try to find out how many of these studies are going on various places and provide some centralized repository for the information. So it's not just a bunch of individual studies, but some compendium.
Yeah, that's a good idea. We could put out an email to, you can start with a broad announcement. We can even seek NSF's help. I think that's a great idea. But to follow up, Simon, just a bit, even though students are not there, I think at least UVA and VT have agreed to open their campuses. And I think as they open in the first few weeks, we'll see a huge sort of thing again. So that's why
I think we don't know yet whether Princeton is going to open in the fall or to what degree it's going to open, but certainly a lot of campuses will, and we should be monitoring that. We should also be in touch with Eric Lander at MIT, who's leading a big effort in the Cambridge.
That would be great.
Yeah, this would help administrators. Maryland is doing a phased opening. Graduate students can go to the lab if they're doing priority research, particularly if they're doing any coronavirus research, they automatically can. But they have a process where they have to fill out forms and so forth, and make sure that they don't have symptoms, etc. But I do think it would be a good idea. In fact, it would even be good to start it now when there aren't many people on campus to get a baseline.
Right. And essentially set up the system in place, I was thinking, right? Because once people come in, we would be ready rather than doing it at that late hour, basically.
One of the problems is going to be what impact the returning students are going to have as transmitters to the communities too.
Yes.
So I agree. It would be good to do those surveys now, and maybe, as I said, to hook up with these other groups that aren't part of the project.
So I think I will follow up and figure out a way to reach out to a broad community. I think everybody here, they can help, I think we might be able to put out a fairly broad email list. It almost suggests to me, Simon, Rita, and others, that just like folks built out a sensor system for earthquakes right after the tsunami, what we have is between individual population movements that we can get from places like Google and other units, and environmental data that Rita alluded to and others, I'm sure, as she said, have collected, and sampling of water system. We have built a multi-sensor capability, or at least the first discussion of a multi-sensor capability to track all sorts of different diseases, because between these three, we have really strong signals for a range of diseases. And that would be a wonderful contribution of our expeditions project if it is successful. Do you agree?
I fully agree. Yeah, great idea.
Yeah, because it provides ground truth to the predictions that we're making, and we're able then, I would think, to verify or solidify the predictions that we do make.
Great.
Anyway, so you got to some of this while I was trying to do this, but it has to do with the geospatial distribution of mutants, and so at the moment with COVID-19, we're talking about a virus, but the general problem with respect to all these things is probably interesting but with viruses in particular, because of their rate of mutation and so on, I think it's interesting with the coronaviruses, they tend to be pretty stable, and people are, of course, tracking, different mutants and placing them and things. But I think there's very good reason to believe there could be some unusual kinds of variation. And specifically, what I think might be an interesting biocomplexity issue to look at is how the distribution is affected by mitigation and other, social behavior and even therapeutics, and when it comes time, vaccines, how it stresses these organisms, and how differences in those things have something to do with what these mutants are. And I know that sounds a little strange, but we actually have some evidence for it, this is not the right place to go into such a long conversation, but I just want to say there's some reason to believe that you can look at measures like transfer entropies and measurements between different layers in these networks, social networks, and the genomics, and actually measure information flows. And they're not symmetric, as you might imagine, upward and downward. If you want to view social and biomolecular as upward and downward, these couplings are not necessarily symmetric. And so I think it'd be worth thinking about that. I think it could give a really different view of how to think about what we're dealing with when we have something like one of these big pandemics. And it's obviously not changing the basic conceptual constructs of how these things are, but could add an incredibly rich and interesting layer. So just to say that again, the geospatial distribution of mutants, or the evolutionary history, however you want to look at it, as a function of things like the social reaction to the presence of the virus. I think this could be a really, really interesting thing to do. The other thing I just wanted to say, as far as the school things are concerned, I didn't think of it until you were just talking about it, and then I'll be brief, is that there's some recent information with military recruits. And it's my understanding that it's at Fort Hood and Fort Leonard Wood, but I could be wrong about that, where they tested all the recruits going into a basic training environment. And in 3 weeks, they had some recruits sick. They tested them, and 30% of the recruits were ill. And these are isolated, they're in barracks and so on, but they're in contact with sergeants and other people that are connected to the world. And it's something of probably a cautionary tale, and also an interesting experiment on exactly what it means to take young people from all over the country, put them in one place together for a while, no matter how isolated and no matter whether you've tested them negative or not, and still experience an outbreak.
Those are absolutely wonderful suggestions, and I do think we need to incorporate them in our thinking. We're doing a study now, working with a medical team. There's some suggestion that there are even variations that occur in the virus as the disease itself progresses in an individual. In other words, the potential that the individual who succumbs, the virus may be slightly altered from the initial. There's just really preliminary evidence, not ours, but some that we've read, And we're doing some studies now to see if that in fact does actually happen, that there would be a change. And it might be selection. It may be that there are variants, mixed variants, that are involved in infection. So just what you're suggesting on a larger scale, grander scale, really makes good sense. And I definitely think we should pursue that from the beginning rather than try to patch it on at the end. So thank you for the comments.
Can I add one thing? I ask one extra question. Then these long-lasting fecal presence of the virus in fecal matter, this is a wonderful opportunity to study exactly what you just said. It would seem to me over the course of a very long period within an individual, whether they succumbed or not. It would be really interesting to look at evolutionary processes in the course of the disease in an individual using those long. I had no idea that they lasted that long. I don't know how I missed that, but that's news to me. That's very interesting. Thank you for showing that.
Well, one thing that I'm fascinated by is the metagenomics of the individuals who are asymptomatic but carry the virus. What are the other microorganisms present? So we're just launching that study. Compared to the metagenomics, what are the other organisms present, as well as other variants, of those who succumb to the disease? And for those who have very severe symptoms but do survive. I don't think this virus operates totally all by itself. And it would be very interesting to see what else is going on. Just as in the 1918 influenza epidemic, there were other things happening as well. And we now have the tools to be able to parse this, to pick apart the components and find out what else is if any, other microorganisms are kind of hitching a ride with the coronavirus.
Thanks.
Any other questions? Maybe one more question and then we can end the meeting. This has been just amazing discussion so far. I almost see, Rita, I can almost think of it as a funnel right now in my mind, where you have a person at the core of the funnel, and the top you have, it's like an hourglass actually, maybe that's a better way to think about it. So person is at the sort of middle vertex, and then you go up in terms of, variables that you have, social networks, environmental variables, and such. And then once you go within the body, you talked about two different aspects already. Chris talked about it, and this sets of hourglasses that are hooked together into a form that really gives rise to biocomplexity is of great interest, I think, so to all of us, and we should maybe pursue this in some form as we go along.
Terrific, I agree.
Yes, Rita, thank you so much. That was really fascinating. One of the things that we're interested in looking at, and I wondered if you had any insight into that, is how co-infection with other enteric pathogens might change fecal shedding with COVID, and whether there might be an interaction there, either the persistency or the degree of shedding. And then that would also maybe be coupled with, the sort of a synergy between those communities and how they affect those dynamics.
That's very interesting. I hadn't thought about that. That's fascinating. Sure, if it was coupled with the presence of Vibrio cholerae, it would sure have an effect on the shedding. Well, there are many others. There's the norovirus or the rotavirus, for example. Yeah, I think it's important to know what other viruses and what other pathogens are present, which is why we're doing and select samples. It's very expensive, but with the technique that we've developed, we can determine that, and I do think it's going to be interesting. In fact, we have a paper with a team at Johns Hopkins, which we have submitted for publication, which is a kind of preliminary analysis of the metagenomics of the in this case, it's the saliva and sputum.
Well, I think that's fascinating. I think one of the things that I would imagine that cholera and the existence of another pathogen that's causing enteric damage is going to increase pathogen shedding of other organisms if they're there. And also what actually creates the mechanism. So what facilitates fecal shedding of the virus in some patients and not in others? Is that something to do with the lung-gut axis, the microbiome? So all of those issues I think are really interesting. And then you throw in the mix other organisms that are already changing the gut microbiome that might then influence the lung microbiome and then change the course of disease. So the diarrhea COVID interaction to me is very interesting, and whether or not there is, and I'm sure there will be pathogen specific influences versus just syndromic influences, I think is a critical area. Hopefully something we're trying to do here in Botswana, and on the one hand, hopefully we never have enough cases, to affect the country. On the other, that would limit our ability to really ask any strong questions. So that's an interesting
dilemma. Because that would be another aspect of being a super spreader.
Exactly.
And that's interesting.
And has anybody been looking? I'm really fascinated with that, the influence the gut-lung axis may have on the way in which COVID presents.
That's interesting, because what we were looking at with respect to the saliva and sputum is to see whether there was a respiratory infection that would facilitate or enable or be enhanced by the COVID virus being present as well. And so that's
right.
Yeah.
Right. And very interesting. Thank you again. That was fascinating.
Thank you.
This has been a very fascinating discussion, Rita. Thanks again. And we'll invite you back again in a couple of months. I think you'll have new results by then. Simon had agreed to do the same. We're trying to have others give a talk, and Golda has been in touch with other folks of the team. So hope to keep this momentum going. Golda, thank you very much. As always.
Yeah, thank you, Madhav. I just wanted to say before people have to hop off that in 2 weeks we'll have another seminar. We'll have Alison Galvani and Abhishek Pandey from Yale presenting. So hope to see everybody in 2 weeks as well.
Thank you again. Thanks, Rita. Thanks, Golda, and everyone else for joining.